Hello, my name is Ketul Popat. I'm a faculty in Department of Mechanical Engineering and School of Biomedical Engineering at Colorado State University. So the research area in my lab is in biomedical nanotechnologies. It's a newer area in biomedical engineering. In biomedical nanotechnology, we actually use nanomaterials for biological applications. One such example would be basically taking materials, changing their properties, making them at a nano scale and using them for regenerating tissues or delivering drugs, or even using them as functional devices inside the body which will cure any disease state.
This is our Biosafety hood. We do all of our sterile procedures in here, so when we're culturing cells, when we're changing our media, we all do it in this hood. So that air actually comes down from the top and then it flows down and out to the sides. That way your samples don't get contaminated with anything else that's in the hood.
One of the research in my lab is in the area of musculoskeletal tissue engineering, especially we are looking at bone regeneration. So there's a lot of mechanical forces that actually act on the bone, and that's why when we want to design a material which would replace this bone, we want to make sure that it has similar type of mechanical properties. So the goal is that we design these materials, we make some sort of a scaffolding system from these materials. These scaffolds would go in the gap where the bone was lost. And then over time, the natural bone which is surrounding that scaffold will start sending cells to this new synthetic scaffold which we have. And over time, the cells in this new synthetic scaffold will start growing new bone. And over the time, we'll see that the synthetic scaffold is going to get degraded as the new bone is actually being formed.
Hi, I'm Barb Smith. I'm a third-year PhD student in biomedical engineering at Colorado State University, working in the Popat lab right now. I look at what the skin cell response is. So there are 3 layers of the skin. There's the epidermis, the dermis, and the hypodermis, which is the underlayer. And biomaterials that interact with each one of these layers have to do it in a favorable kind of fashion. What's happening currently is that prostheses that go through the skin rejects them. And so it becomes an open doorway basically for bacteria to infiltrate through. Now we're finding that we can increase the interaction between the material and the cells. So not only more bone matrix is produced on the outside, but it infiltrates into like about a nanometer into the material. And so you get a better kind of interaction with that.
There are 2 things that I really like about my job. I really enjoy working with students, and being a professor lets me do that in different ways. When I go in the lab and when I work with students, I really feel very energetic to pass on my passion for research to them so that they can continue their research. I also really like working with students in a classroom environment for the growth factor and the peptide which is there. So based on that affinity constant, you can have strong or weak binding, and you can modify. The field of biomedical nanotechnology is very new, and there are a lot of challenges which are there in terms of translating some of these nanotechnologies to real world. But I believe that we will see some breakthrough in biomedical nanotechnologies in the next 10 to 12 years. And once we understand it better, I think we are not very far from actually having an actual therapy which can go into patients.
It's really cool research. We're hoping that it keeps moving in the field.
I hope that some of the work that we are doing in my lab right now would somewhere fit into this big biomedical nanotechnology arena.